Hi everyone, so my name is An Nguyen and thank you for organizing such a wonderful event and thank you for having me here. So I'm a postdoctoral researcher at the Department of Infectious Diseases at Monash University, and today I will present a paper that just recently published last month named AMR-GNN multi-presentation graph neural network framework to enable genomic and antimicrobial-resistance predictions. So first, to give you some impressions of AMR. So AMR is suppressing global text, and then in systematic review at the Lancet in 2024, AMR is now directly responsible for nearly 1.2 million deaths annually, and then if you don't take any actions, the death toll will be up to 2 million by the end of 2050s. And because of that, the governing authorities had identified some of the key issues that need to address to prevent the spread of AMR. And then one of the most important factors is the rapid diagnostic test for AMR. This is because why the gold standard, why the gold standards susceptibility testing method, which is the broad viral dilutions, they are highly accurate. However, the problematic is their turnaround time, and then also they are quite label intensive as well, and it can take like 2 to 3 days to return the susceptibility test, which is in some case of severe infections is not acceptable because the difference in the time can be mean the difference between the life and death. And because of that, there are many methods that are proposed to address the rapid AST testing. And amongst of them, the molecular AST method is one of the promising approach. However, the current approach is the rules-based approach, which is based on the presence or absence of some AMR determinants to determine AMRs. For example, like you can see there's some very famous example, like the mutation in vein A or vein B in vancomycin resistant ephasium, Enterococcus ephasium, or the mutation in MEK in MRSA in methicillin resistant Staph aureus as well. However, these current approach have some drawbacks. The first drawback is that they cannot account for novel AMR determinants where there's no literature showing that. And also, given that they are also unable to learn some interaction in terms of the multiple AMR mechanisms. And you can see on the right side in the Pseudomonas aeruginosa, when the AMR is likely emerged from different multiple AMR mechanism rather than just one single determinant. And because of that, we need a more sophisticated approach, which is not only accountable for the multiple interactions of AMR mechanism, but also able to detect the novel AMR mechanism as well. And then this is where machine learning came to play. So with machine learning approach, one, the model is trained. You can quickly predict the AMR given your input. So in this work, we are focusing on the whole genomic sequencing data to predict AMR. This is because nowadays the cost to sequencing the bacterial genome is now significantly reducing, make it now more data available to train the machine learning models. And then you can easily download the genome sequencing from some public dataset like the NCBI as well. And then this made whole genomic sequencing data a very valuable resource to train the machine learning model. And because of that, we are using whole genomic sequencing data to train the machine learning model to predict the AMR in Pseudomonas aeruginosa. However, we identify there's some gap in the literature regarding to the way that we are developing machine learning models. So the most important thing is that because whole genomic sequencing data is a very high dimensional data, which means that, for example, like with Pseudomonas, a whole genome can contain roughly about 6 million nucleotide sequence, and which means that whole genome sequencing data can be represented in so many different ways. So for example, like the KMers one, we see the substring of the nucleotide, and also its variants called the unitics one, which means that they're trying to combine the overlap KMers into one single but longer nucleotide 6 substrings, just to reduce the computational time. And also you can the whole genomic sequencing data can also be represented in different ways, like single nucleotide polymorphism, which need to compare the sequence with a reference genome, or the presence of or absence of some AMR gene as well. And also whole genomic sequencing data can also present in the form of unimaged like features, such as the frequency cascade representation, as you can see on the right side, where each KMers can be located in a cell in a cell, and then from that they will try to encode for each cell with the frequencies count for each KMer presented in the genome as well. And because of that, in the machine learning models to predict AMR, nowadays we don't have like a consensus, which is the best way that we present the data for the models. And then we identify that gap. And then we think that maybe when we try to integrate different kind of genomic representations, we can hopefully improve the performance of the models. And then it's also showing that the benefit of the multimodal learning. So for example, in a study in 2020 by Kennedy and they are doing they're developing the machine learning model to predict AMR for different type of antibiotics using both genomic sequencing data and then also transcriptomic data. And then on the right side, you can see that except for the ciprofloxacins, for other 3 antimicrobial testing, we can see that the use of multi data modality can help improving the performance of the model. So for example, you can see in terms of the F1 as macro, so we see the pink one here is the present or absence of a gene, or the tag, the live in as well, which is the combination of both gene present or absence, the transcriptomic data, and also the SNP variant as well, can help improving the performance model compared to the model using just single genomic, single data modality. However, one issue with the approach is that nowadays most studies just using some simple concatenations to integrate different modality for the machine learning models, which means that all of the features are gathered into, for example, a table-like data format, and then also, and then after that, the machine learning model, we're trying to understand that. But given that nowadays we're having a more modern deep learning architectures, we hypothesize that maybe using different kind of data modality in different aspects of the model, we can help improve the performance. And the way we're doing that is that we propose AMR-GNNs, which leverage the RAF convolutional neural networks to predict AMR. So the way we're doing that is that the first we select some of the most common genomic feature type to predict the AMR. So here we selecting the SNP, the unitics, and then also the frequency scarce game representations. For the first step, we trying to train the model using just single modalities. And from that, we identify Unitix at the best performance models. I will show you the result in the later slide, but here just want to walk you through the workflow. So we're using Unitix at the node features for the graph. And then from that, we're using the two remaining modality as the auxiliary features used to construct the edge of the graph. And then from that, we are trying to learn two separate graph for each type of edge we have. And then at the final stage, we're trying to combine the embedding layer from those two graphs using layer fusion approach called low-rank multimodal fusions to perform the predictions. So by doing so, so first of all, we're trying to collect the data. So our data sets including 2,515 Pseudomonas isolates collecting from our in-house data from the Alfred Hospital at Melbourne City, Australia. And then also we're trying to collect the data from 9 public datasets. We selecting 2 smallest datasets and keep them at the whole dataset for external validations. And then we are validating our model with 12 anti-Pseudomonas agents. So first of all is the selecting the node features. So as I mentioned before, we are using Unitix, SNP, and frequency scarce game representations. We also trying to different kind of models for different genomic representations. So which is ElasticNet for Unitix and SNP, and for frequency scarce game representations, we're using convolutional neural networks. Is because this representation is more like an image, and then we using convolutional neural networks with a very suitable for image data. And from that, you can see in terms of AU, as you see, the Unitix one is actually the best genomic features for all of the tested data. So, and because of that, we're choosing Unitix as also as the model trained with the Unitix one as the baseline models for comparing it with our AMR-GNN models. And by using the Unitix models at the baseline ones, we can see that AMR-GNN models with the one with the orange line here. So the baseline model is actually the one with the pink line, as you can see. So in most of the case, the pink line is inside the orange line, which means that AMR-GNN models actually outperform the baseline model. So in like 11 out of 12 tested antivirals, and then for the remaining antivirals, we are achieving a comparable result. So the AUSC performance is actually pretty good. So the lowest one is the self-esteem, which is 0.81, and then the highest one is topramazine, which we are achieving the AUSC at 0.97. And further than that, we are also showing that AMR-GNN model, they also able to mitigate bacterial population structure, which is a very common issue when you trying to developing machine learning models to predict AMRs. This is because for bacterial genome, the child genome is actually inherited from just one single parent, and because of that, it's a very high chance that some non-causal mutations are likely co-inherited with the causal one, which means that it makes it very difficult in sometimes very difficult to identify what is the variants that causing the AMR, or even that some model they are just trying to remember the lineage information to predict the AMR rather than trying to use the actual causal variants to predict AMRs. And because of that, we propose decouple AMR-GNN, which means that we're just trying to remove the edge connecting the node within the same sequence type. So by doing so, we are forcing for each node, it's just only trying to aggregate the information, the genomic information from the nodes that not in its lineage, and then hopefully it can have the model to identify the correct causal variant for AMR. And by doing so, we think that we are actually slightly improve the AUC improvements in all of the tested microbials. As you can see here, the gap between the AMR version, original version, and then the gap with the AMR-GNN decoupled version is actually not very significant, but we did get some improvement with our decoupled AMR approach. And to validate our AMR-GNN model, as further than that, we're trying to perform some benchmarking study with some approach that are tailoring for Pseudomonas resistant isolate. So the first approach is we choosing a rules-based approach with trying to curate a very large set of AMR determinants from the literatures and using that to detect the AMR for Pseudomonas ones. So the second approach is ARDABs, which using both the rules-based determinants and also then trying to identify some additional variants learning from the machine learning models to predict AMR. And then finally, it's the VA MPR, which is a poorly machine learning model to predict AMRs. And then in most of the case, for the internal datasets, our AMR-GNN models actually outperform one of the benchmarking approach for first in terms of the F1 score. However, with the external datasets, why the majority, why AMR-GNN models is doing better than others approaches. In the majority of cases, we did acknowledge that for some cases, such as like the monoponeme ones, actually AMR-GNN models actually perform worse than other approaches. And finally, we are validate our AMR-GNN models using others pathogens gathered from the BV-BRC dataset, which including E. coli one, the E. coli, the Clade pneumoniae, the Ephesium, and the Staph aureus. We are collecting more than 23,000 genome from the BV-BRC dataset with different susceptibility testing related to each species. And then, so on the middle panel, you can see is that actually the AMR-GNN achieved a very good performance for in terms of AUACs. So in all of the case, we are achieving more than 0.9 AUAC, except for the piperacillin tetrabactam in E. coli, which is when the AMR-GNN achieved the AUAC at 0.87. And finally, we are also trying to compare our AMR-GNN models with universal AMR prediction tools called the REST Finder. The reason that we choosing REST Finder is that actually in a benchmarking study conducted in 2024. Actually, Respider is the best AMR prediction tool out of all of their tested approaches. And then, as you can see here, for the result, AMR-GNN actually perform better than rest finders in most of the case, except for some cases. So, for example, like the fusidic acid in Staph aureus or the terramycin in E. coli. So, with the limitations, so first of all, we are acknowledge that the use of the second type to account for populations structure bias is actually a bit more simple. So in the future, we might try to use the core genome MLST to better addressing the lineage bias. And also, we're thinking that we need to perform the prospective study to assess the AMR-GNN utility in guiding for the real treatment decisions. And finally, for the unique database, when we are acquiring more genome in the future, we may need to update it. And then it may raising some of the computational complexity as well. So this is my talk, and thank you for your listening.